Hello everyone, welcome to EduRev. In today's session, we will talk about Mendel's laws of inheritance, which is based on Mendel's hard work of several years in form of laws. So, let us take more of his creations in detail. In the previous video, we talked about how Mendel observed 7 contrasting traits in garden pea plants, and that were seed shape, seed color, pod shape, pod color, flower position, and stem length. And we also learned what monohybrid cross was, where Mendel took only one character of a pea plant and crossed two pure tall and pure dwarf plant. And in this we saw that the first generation was all tall plant. In the monohybrid cross, we saw that the first generation was all tall. But after selfing the first generation, that is capital T small t, and again capital T small t, now the F2 generation showed one dwarf plant as well, which gave us various phenotypic ratio and genotypic ratios, in which we saw that the phenotypic ratio the phenotype we talked about was 3:1, and genotype that we saw in this case was 1:2:1. Now phenotype is what you can see from your own eyes. So when we saw the second generation, we saw that the 3 plants were tall and 1 plant was dwarf. But when we looked at its genotype, its genetic nature, so we saw that 1 plant was homozygous tall, 2 plants were heterozygous tall, and 1 plant was dwarf. So yes, till now this is what we know about the monohybrid cross done by Mendel. Now based on this cross, he gave 2 laws which we are going to discuss in this session. Before studying about the 2 laws of inheritance, do you know why Mendel selected garden pea plant for his experiments and not any other plant? It is because pea has many alternative traits, and because of which it is easier to study each trait of what Mendel has observed. So Mendel observed 7 traits in pea plant which he studied separately and then given out some results that we study today. It also produces large number of seeds, because of which it is very easy to grow this plant because of obviously the number of seeds it is producing. And lastly, it completes its life cycle in just 1 season, due to which studying about its progeny is easy and does not take a lot of time. So finally, let us start with our first law, that is the law of dominance. That is the law of dominance. Now, what do you mean by dominance? So dominance simply means to dominate. If something is dominating on the another thing, that is what is law of dominance in simple words. But in words of Mendel, let us study the law of dominance, which says first of all that characters are controlled by discrete units called factors. So what Mendel had given was not about alleles, not the term alleles, but he called those characters as factors. And what are the characters we are talking about? For example, we are talking, because now we know that those are called as alleles, so I would rather write down here as capital T. Now this is the particular character which shows the tallness of a plant. So this particular T, he called it as factors. And the second part, he says that factors occur in pairs. Means I can write down here capital T, because factors occur in pairs, and small t for the dwarf plants. Means it is for the tall plant and it is for the dwarf plants. Now for the third part, he says that it in dissimilar pair of factors. First, we will find out that how dissimilar pairs are occurring. So they occur because of the gamete formation. And in the gamete formation, one gamete comes from male and another gamete comes from female, as we have studied earlier also. So one capital T will come from male and another small t will come from female. So the plant will become tall in this case also. But here this has now become a dissimilar pair. This is the dissimilar pair we are talking about. Now study the third part. In a dissimilar pair of factors, one member of the pair dominates the other, which is the recessive one. Means this capital T is dominating over the small t. And that is why the result of first generation is tall and not dwarf. Because the factor tall is dominating over the recessive one, that is small t, that is for the dwarf. So that is what is law of dominance. So according to the law of dominance, the definition is that in the heterozygous individuals, remember we are talking about heterozygous individual and not homozygous. In homozygous we cannot find law of dominance because they both are equally expressing. They both are equally expressing. So that is why we are taking a heterozygous individual, or we call it as hybrids. So the character is represented by two contrasting factors. Now two contrasting factors are what? Capital T, tall, small t, dwarf. These are the contrasting factors, which are called as alleles. Now let me remind you what alleles are. For example, this is the gene. This is a gene. Now this gene is for height. It shows the trait of a height. Now alleles basically are the two contrasting pairs which actually defines the phenotype. This is tall. This is the dwarf. Now the traits which are showing tall and dwarf, two contrasting pairs, are the alleles which are on a gene. Yes. So we also discussed about the concept of dominance, that how any allele dominates. And if you remember that we talked about that how, for example, this is the chromosome. This is the chromosome. Now we talked about how mRNA is produced from DNA and how it produces proteins, if you remember. Yes, how it produces proteins, which gives either tallness or dwarfness or any character you are studying about. And how, on the other hand, we studied about a faulty DNA, which gives us faulty mRNA, which further gives us the faulty proteins, and which gives a dwarf plant. So this is what concept of dominance is, and that is how any factor you see dominating another one is because of this concept only. So Mendel gave us law of dominance. But here is a fact, that evolution is a process that is happening for so long and will keep on happening till the earth we know is intact. So because of this, few exceptions were later on seen to the law of dominance, and those were called as the incomplete dominance and the codominance. Now what do you understand by incomplete dominance? It simply means, if we talk about simple English, it simply means the domination which is not complete, the dominance which is not complete. The dominance which is not complete. Simply, it means incomplete dominance. And how incomplete dominance will take place? Because in concept of dominance, we have seen that how a particular allele is able to dominate the another allele. But here we see the term has now become incomplete dominance. How come this is happening? So let us suppose that this is the capital T and this is the small t small t. Now it is not able to produce enough protein. Not able to produce enough proteins. So that is why it will remain mutant. Yes, proteins as we have studied. Now, for example, this capital T is able to produce 100% of proteins. Now, when the crossing takes place, now according to the gamete formation concept, one capital T will come from male and small t will come from the female. But here, at this point, due to some genetic variations, due to some genetic variation, this capital T is not able to dominate the small t now. Why? Because now it is only producing 50% of proteins and not 100%. And this is the mutant. This is mutant. So it is also not producing enough amount of proteins. And whereas capital T was to produce 100%, but it is only producing 50% of protein. So now the dominance will not be complete. The result will come intermediate. The result will become intermediate. Yes, so that is why it is called as incomplete dominance, because the result in dominance is never intermediate. It is complete domination. But here it is incomplete because of the intermediate results are coming here. Now coming to the codominance. Now what do you mean by codominance? Codominance means that, for example, two people in a room. Let's suppose this is a room and here is a person A and this is a person B. Now these two people are in the same room, but they are different. This is A, this is B. But they both are able to produce 100% of proteins. Impressive, right? They both are able to produce 100% of proteins. So that is why they are expressing equally, but not dominating. These are not dominating on each other. Can you see that? Because they both are producing 100% of proteins. So they are equally expressing but not dominating. But what was our law of dominance again? That one dominates over another. One dominates over another. But this is not happening in this case also, and this is not happening in this case also. So that is why incomplete dominance and codominance, they both are exceptions to the first law. And now we are going to study in detail about incomplete dominance and codominance with the help of examples. Let us now begin studying incomplete dominance. So incomplete dominance is a form of intermediate inheritance. So now the word has arrived. Intermediate. There should not be any intermediate in dominance, but it is there since it is incomplete dominance. So a form of intermediate inheritance in which one allele for a particular trait is not expressed completely. The trait is not expressed completely over its paired alleles. Means one allele is not able to dominate over the another one. That is what is incomplete dominance. And the example of incomplete dominance is the snapdragon or the antirrhinum species. So this is very, very important example, very, very important topic, incomplete dominance, and the question on its example can come where if you'll find these options, snapdragon or antirrhinum species, you have to simply tick on it. All right. So this is the example of incomplete dominance, the snapdragon or antirrhinum species. Now, what kind of incomplete dominance was seen in this case? So here they have taken the flower color. So the red flower color in case of snapdragon, that is capital R, is dominant. It is the dominant red. Red color is dominant. And whereas the white color flower is recessive. The white color flower is recessive, taken as small r. Now, when they crossed it, the capital R and small r, the F1 generation was which color? Which color can you see on your screen? It is pink color. But according to the law of dominance, it should have been red, right or not? Because here, dominant is red. So in F1 generation, it would have been red, but it is not. It is now pink. All F1 generation is pink. And why? What is the reason behind it? It is because the red after crossing is not able to produce enough amount of proteins. So that is why the complete red color is not visible in the F1 generation. Instead, the pink color is seen in the F1 generation, as you can see in this image. Now let me draw F2 generation to see what are the genotype and phenotype in this case. So I'll draw the Punnett square. And what do we do in F2 generation? We do selfing of F1 generation, if you remember. Selfing of F1 generation. So these are the F1 generation. So let me take these two flower, capital R, capital R, small r. Now I'm crossing it. So I'll cross it as capital R, and capital R, small r. Now this is capital R, and these two are small r. Now let us see that what is the phenotypic ratio. Phenotypic ratio. If you can find out in your mind that what can be the phenotypic ratio. So you will think that it might be 3:1 because 3 are dominant here. But remember that we are studying incomplete dominance. So if you think that it will also come in 3:1, then it will not, because visibly also, phenotype is what? What you see visibly. Visibly also you will see them as pink. Yes or not? You will see it as red. You will see it as red. And you will see it as white. So visibly also you are able to see 3 colors. So one is red color flower, 2 are pink color flower, and 1 is the white colored flower. So that is the phenotype, which is same as genotype, because if you are seeing genotypic ratio as well, so you will also find that 1 is the R red pure dominant, 2 are heterozygous dominant pink, not dominant, intermediate pink, and 1 is the recessive white. So in this case, phenotypic and the genotypic ratio will be same. And here, no dominant red color is coming, but the intermediate pink is being formed, and that is the reason it is called as incomplete dominance. So now let us talk about the codominance now. So codominance is a heterozygous condition. So I am repeating time and again that we are only talking about heterozygous condition and not homozygous, because the exceptions, the conditions we are studying is not that much applicable or not applicable on homozygous conditions, but only on the heterozygous conditions, in which both alleles at a gene locus are fully expressed in the phenotype. Here we are talking about the full expression of both the alleles, in which one allele is not dominating over the another, but both alleles are expressing equally. So the example of codominance is generally seen in the human blood groups. Yes, you will find the exception of law of dominance in the human blood group. Now see this flow chart on your screen. Alright, it is not very clear till right now, but after discussing, it will be very clear to you that what actually it depicts. So here, for the blood group, we have taken the symbol as I. One thing. Alright, capital I. Alright. Now, for example, if a person is having blood group A, so the antigens which are present on the blood will be A. These will be A, all over A. And if a person is having blood group B, so on the blood, the antigens which are present will be B, capital B, capital B, capital B, all over the blood. And what if the blood doesn't have any antigen? If the blood does not have any antigen, then what? So the blood group in that case is O. So you might have seen that some people have blood group O as well. It is because there is no antigen present on the blood, neither A nor B. So that is why that blood group is called as O. Now let us talk about how the blood group or how the blood group of a person is decided. Now look at the first one. Now here we are talking about blood group A here. So you see that since the alleles occur in pairs, so I can write down that capital IA, capital IB, or sorry, capital IA again. So this represents which blood group? It represents A blood group, because in both the genes, both the alleles are capital A. Both the alleles are capital A. That is why it is representing as blood group A. But this is the homozygous dominant. Homozygous because both the alleles are same. But there is one more condition in which a person can have blood group A. That is, if a person has capital A as one allele and one mutant allele which doesn't have any antigen. Yes, that is capital IA or small I. So in this case, this capital A is dominant over the small I. Yes, so that is why the blood group will become A in this case. In both the cases. Here you can see IA or IA or simple I or small I. Yes, it can be written as small I also. You don't have to get confused. It can be written as small I also, and it can be written as like this as well. Both the situations are right. Now if we talk about blood group B. So here IB and IB gives blood group B. Or in the heterozygous condition, IB or small I will also give blood group B. Now, where is codominance lost in this case? Because both the conditions are dominating. The IA is dominating over I. B is also dominant over the small I. But we are talking about codominance here. So, in some cases, in some humans, you might have seen that a person is having a blood group AB. So, it can be written as IA and IB. Now, both are dominant, but they are not dominating on each other. So, that is why this condition now is codominance. This is the condition which is the codominance condition, where A and B are equally expressing. They are equally expressing, but not dominating. Yes. So this is where the codominance is present. And in the last case, where there is blood group O, here there is no antigen which is present. It is mutant, and That is why the blood group is O in this case. Yes, you can see II. Now, if you see the red blood appearance also in the middle, so you will find that they have used a triangular shape for antigen A, a circular shape for antigen B. So you see that on this blood, all the triangular shapes are showing the antigen A, all the circular shape in this case are showing antigen B. Yes? But in case of codominance, you will find that triangular A is also present, circular B is also present. Triangular A is also present, circular B is also present. Here also triangular A is present and circular B is present. Now this is the codominance. They are existing, expressing in a similar way, in the same manner. Both are strong, both are expressing themselves and not dominating each other. Now this is the case, this is the example of codominance. Did you notice that example of ABO blood grouping is also providing a good example of multiple allelism? Now multiple allelism, what do you mean by multiple alleles? Multiple, we are studying about multiple alleles. And where we have seen it? We have seen that 3 alleles are governing the same character, that is blood group. Yes? So antigen A is governing blood group A or giving out blood group A. Allele B is giving out blood group B, and allele I is giving out blood group O. So this condition where 3 alleles are governing the same character is called as multiple allelism. Although the characters are controlled by 2 alleles in an individual, remember that, but more than two alleles in a population. Means if we have to look for multiple alleles, then we'll study the whole population. But if we are studying an individual, then we will only see the characters which are shown by 2 alleles. So it is a slight difference, but multiple alleles, now you understand that where multiple allelism is seen. It is seen in the population. So let us study about one more condition, that is pleiotropy. Now what is pleiotropy? So pleiotropy, in pleiotropy what happens is a pair of allele, it controls more than one character. In earlier topic, we have seen that 3 types of alleles are controlling only one character. But in this case, a pair of allele is controlling more than one character. So this condition is called as pleiotropy. Now see this image in front of you. You see a grain of pea in this case. So let us talk about the characters, the various characters we are talking here. First one is the round shape of a pea. Which is dominant? Round shape. This is the round shape of a pea. Got it? Now, let's talk about the second character. Why the pea has a round shape? Because of what? It is because of the content present inside it, and that is starch. So starch synthesis is the another case. Starch synthesis is the another case. So let me draw some large grain of starch inside it for you to understand that the shape which this pea has attained is because of the grains of starch inside it. These are the grains of starch which are present inside it. Yes? Now let us talk about the recessive pair of alleles, that is small b. Now they are not giving round shape, as you can see, they are giving wrinkled shape to the pea. And let us talk about the second character. Why the shape of this pea is wrinkled? It is because of the small starch grains inside it. So let me draw some small starch grain inside it. They are not as big as you can see from the dominant ones, as it is recessive. So I'll draw the small starch grain instead. Now coming back to the third condition where you see the heterozygous dominant condition. Here capital B is dominating small b. In a way, yes, we have seen it. So the shape is round because of it. Round shape. But now can you make out any difference between this purely dominant one and this heterozygous dominant one. You see that the small is a bit smaller as compared to the dominant one. So we can say that this is an intermediate condition. And since it is an intermediate condition of a P, then what type of starch grain will be there inside it? Will it be smaller or larger, bigger or smaller? It won't be bigger, it won't be smaller, it will be intermediate. Intermediate starch grains. Starch grains. Yes? Since this term intermediate has come, so do you know why this condition has appeared? This middle condition, why it has appeared? I hope some of you have guessed it right. It is because of the incomplete dominance. Incomplete dominance. The intermediate term, wherever the intermediate term comes, it is because of the incomplete dominance. Got it? And the two characters we have studied in all three conditions is what is pleiotropy, because they are all controlled by a single pair of alleles. Let us now start with the second law of inheritance, that is law of segregation. Law of segregation. Now what do you mean by segregation? Segregation simply means to divide. To divide. And also one more important thing, that the second law, that law of segregation, is known as a law with no exception. No exception means this law of segregation do not have any exception that we know of till date. And this kind of question can also come in the NEET exam as well, that the question will be asked that name a law with no exception. Then you have to take the second law of Mendel, that is the law of segregation. Now let us see what this law states. So, every individual possesses two alleles of a gene. We know this, that two alleles are possessed in a gene. And these alleles segregated. These alleles segregate. Segregate means what? Divide. Segregate from each other during gamete formation. I hope you all know what gamete formation is. So let's see. Capital T, for example, tall plant. And small t, that is the dwarf plant. And since we know that why we write capital T and small t is because the two alleles occur in pairs. Now, what happens in law of segregation, what Mendel has told, that the alleles segregate during that only one gamete is transferred in the offspring from each parent. Segregation, they divide. Yes, and the small t will come from this parent. Getting my point? So this is what is segregation. So these are segregating from their parents. The gametes are segregating from their parent plant and then coming into the offspring. Also, these alleles do not show any blending. They do not show any blending. What does this means? What does it means to not show any blending? It means if you see that this capital T is not getting merged with small t. Do you see this kind of condition? No. It means the capital T and small t are existing with each other, but this capital T is dominating over the small t. So that is why this plant is tall, but there is no blending. There is no blending. There is no mixture of these two gametes. Yes? So I hope you understood what law of segregation is, that here the gametes are segregating and then coming into the offspring. That is what the law of segregation states. So let us try doing a simple question to check our knowledge. So the question says, in the snapdragon plant, the appearance of a pink hybrid, capital R, small r, Between a cross of red, capital R, and white, small r flower, parent indicates. What does it indicate? What does it indicate? The appearance of pink hybrid plant. It indicates what? So it indicates incomplete dominance. Incomplete dominance, which is seen in which plant? Snapdragon plant, as we have discussed. And why? Because if you cross capital R, that is red flower, with the white one, small r, with the white one, so the intermediate condition in this case after gamete formation is pink. The pink flower is formed in the F1 generation, which is the intermediate condition. And that is why this condition is called as the incomplete dominance, which is the answer. So yes, this was all about Mendel's law of inheritance. I hope you people have understood about the two laws and some exceptions to first law. You can now go to EduRev and attempt its respective tests to understand the topic in depth. You can also unlock all the logged videos, docs and tests of class 12 with EduRev Infinity Plan and ace your exams at less than 80 rupees per month. Thank you.